Wominjeka, welcome to Melbourne Museum and the museum's Victoria Lecture Series. I'd like to begin by acknowledging the traditional owners of the lands where we meet tonight, the Woiwurrung, Wurundjeri and the Bunwurrung peoples of Eastern Kulin Nation, and First Peoples language groups and communities across Victoria and Australia that may join us tonight. And I pay my respects to the elders past, present, and acknowledge we are guests on their country. My name is Dermot Henry, Head of Sciences. We are pleased to be presenting here in person at Melbourne Museum to a live audience as well as online. The museum's lecture series seeks to connect the public with the importance the museum's lecture series seeks to connect the public with the important and wide-ranging research of the museum staff and its collaborating researchers. Appropriately, in National Science Week, this evening's lecture, SNAP, CT Scan, Model, Predict, what new technology reveals about rare reptiles and frogs, highlights innovative research being applied to understanding and conserving Australia's unique fauna. Tonight, we will receive a presentation from our three speakers, followed by questions from the audience, including our audiences online. It's now my pleasure to introduce our speakers. Rocio Aguilar, who's a research associate at Monash University and also at Museums Victoria. Rocio is an Argentinian herpetologist who has called Australia home for the past 12 years. Rocio specialises in ecophysiology and morphology, thermal biology and CT scanning in order to create a better understanding of the evolutionary processes which have made Australia the most lizard-diverse country in the world. We also have Juan Valbuena, who's a PhD student at the University of Melbourne and also co-supervised here at Museums Victoria. He's a herpetologist who specialises in computational biology, remote sensing, and graphic information systems. His research has taken him around the world and often involved handling highly venomous snakes. He combines his passion for exploration with machine learning technologies to uncover the beautiful diversity of the planet and understand its relationship with the environment. And our third speaker tonight will be Rebecca Rose. Rebecca is the Assistant Curator of Herpetology at Museums Victoria. She has studied a wide variety of animal species, both in Australia and Africa, including reptiles, mammals and birds, and she has worked in the areas of ecology, genetics, conservation and animal behaviour. So please make them welcome. And Rocio's going to kick off, I believe. Yep.
Thank you so much, Dermot. And yes, hi, I'm Rocio, and thank you so much for being here, the ones that are in person and the ones that are online. I hope you enjoy our talk today. So, again, quickly, I will repeat Dermot's greetings that here at Museums Victoria, we want to acknowledge the Woiwurrung, Wurundjeri and Bunwurrung peoples of the Eastern Kulin Nations where we work and meet today, and First Peoples language groups and communities across Victoria and Australia. We pay our respects to their elders past and present. We extend that respect to the Aboriginal and Torres Strait Islander people here today. Excuse me. Okay, cool. So let me introduce you to the team. So this is a team led by Dr. Jane Melville from Museums Victoria Monash University and Dr. Jay Black from University of Melbourne and Dr. Scott Hucknall from Queensland Museum, and a whole bunch of us, including me, Beck, and Juan sitting here, and other panel investigators are part of this amazing project that involves, well, actually is focused on trying to use the huge amount of fossils that we have here in Australia. So what we are trying to do is to use existing paleontology collections and innovative technologies to try to understand how the quaternary period, and in brackets, Juan is going to explain what quaternary period means. That's not my job. And so what the quaternary period back then, how it shaped our present and past frog and reptile communities here in Australia. And the other really cool stuff that we are going to do within this project is that we're going to do that all along 3,000 kilometers on the eastern coast, on the eastern part of Australia. So that is a very, very like a massive, large scale project that we are going to get into. And so what our research questions? So what we want to know is how has Australian reptile and frog biodiversity changed over time? That is one of our first questions. But that, when we say over time, we mean in the past 500,000 years. So that is a very, very broad period of time. So we are going to work in this project in a very large scale in terms of time and space, both. And we want to know who and where they are. So these species of reptiles and frogs. And the second thing we ask is how is climate involved in all this? So do these changes correlate with environmental changes in the past? So that how did affect it all the morphology that we see and biodiversity that we see nowadays? And the last thing we are gonna ask ourselves here is, well, can we predict what is going on, what is gonna happen in the future in terms of how species are gonna evolve and how the environments are gonna be in the future with all the information that we have from the past, the present, and how we model the future. So that's what we want to know. How are we going to do this? So the way we are going to do this is to compare fossils to present day animals. So we are going to use 3 broad innovative techniques. And the first one is going to be like the 3D CT scanning fossils, and that is 3D imaging technology that I'm going to talk about later. But mainly what I want you to remember is that from these, what we want to assess is to quantify shape and size to be able to compare with the fossils that we have and the present day animals that we have. The second big method that we are going to use is ancient DNA, and Beck is going to be talking about genetic stuff later, and Juan is going to be talking about the ecological modeling. That is the third methodology we're going to use. That is computer-based technology to understand species distributions and community assemblages changes. Okay, so I say that we are going to compare fossils with present-day animals, but fossils from where? Ta-da! From caves. So we have so many caves and not available. They are there. Yes, they are available here in Australia. So so many different climates that they cover. So here in Australia, like our majority of fossils come from caves. And why? Why is that? It is because caves provide an amazing, good environment to preserve fossils throughout a long period of time. So, So we have many different study sites across Eastern Australia and a little bit in South Australia too. And these different sites represent different cave sites, and they belong to back in the Pleistocene, how the climate used to be, and now that's what we are offered with different kind of cave shaped by the Pleistocene climate thing. So, for instance, the Chilango caves in Queensland, they are kind of savanna tropical kind of climate. Then we will assess our other study site would be in Mount Etna, well, east, because we already have deposits from there. So from all of these sites. So Mount Etna, that is just subtropical, and Cathedral Cave in New South Wales is a kind of a warm temperature with a continuous rainfall. So imagine kind of a sauna kind of ambient, and the other two there. So a more temperature with just winter rainfall regime. So and before I leave Juan with us to talk about the Quaternary that I mentioned before, because that is the period we are gonna run our study about. So I'm gonna just give you a snapshot of what we do when we extract fossils. So paleontologists go there to the caves, they extract like big chunks of dirt with bones. We put it in, they put it, I assisted in one of the expeditions, which it was a privilege. So we put it in the depository, and then full big trays, like hundreds and hundreds of trays, go back to the lab. And that's where we extract the organic material from the bone, but we also can extract, if you can see there, Nicole, that it was your ring. And so if you can see that, how tiny that is a mandible of, I think that is a skin. No, that is an agami, a little dragon. So they are tiny, but you can still see them. So sometimes we just pick them by hand, and then we fill on all these trays, and they go to the museum to be analyzed and ID'd. Okay, so now Han, let us know what happened with our past climate that created all these.
Yeah. Hello, everybody. Yeah, let's talk a bit about the Quaternary. Although maybe here not everybody has heard about the Quaternary before, but what if I say the Ice Ages? I guess everybody here has watched the movie Ice Age, and this movie represents a time that started 2.5 million years ago, and it's a frozen world dominated by big mammals. But here, the picture in Australia looked a bit different. So it was more like a no frozen world dominated by big reptiles. But also there was not only one climate during all this period, but there was cyclicity between different climates, cold and dry climates that represent here more desertic areas, so expansion of desertic areas, warm and humid climates that they are more like expansion of tropical forests, passing by intermediate climates like mesic forest, that the one we can see here in Grampians National Park. And this cyclicity was due to the movement of the Earth, the energy that reached the planet from the sun. And since 800,000 years ago, approximately, this cycle restarts every 100,000 years following the eccentricity. Basically, the orbit of the Earth varying the distance to the sun every 100,000 years, and we had different energies reaching us. So let's see the climate here in the present, and here we're going to be focusing mostly in the warm areas, in the warm colors, that represent the arid climates. And if we look back at, if this works, yep. If we look back 20,000 years ago, for example, we're going to end up in a cold and dry climate, more desertic area. And here we can see in the East Coast how the arid climate has expanded toward the coast. Similarly, is going to happen in the prediction that we have for 2100 after climate change has impacted us a bit more, right? So if we get a point in the East Coast over the time, and we consider two scenarios to make it a bit simple. If we have a species of lizard living in the present in a mesic area, but we didn't find it in the past, it's maybe because it was suffering, maybe was too hot for it. But this information tells us that this animal is likely not to like this environment and probably would choose a different one, right? So for us as a scientist, that can tell us that in the future, probably it's going to dislike a habitat like this. We can consider a second scenario in which actually our animal enjoy the hot temperature and it's just like having fun there, right? And in this case, it's telling us that an animal like this is likely to do it all right in the same type of habitat in the future. Our study approach to study how the changing climate impact animals here in Australia is called macro-ecology. And in macro-ecology, mostly we try to find the explanation for ecological patterns by finding a statistical relationship between the components that explain that pattern. But let's make it a bit easier. Imagine that we want to find the distribution of this frog, right? So trying to find a statistical relationship between the frogs and the climate that explain the distribution is basically a macroecological approach. In the past, in terms of the distribution, well, sometimes it's still used. There's another methodology that is just to draw a line around the records of the species or where they were found. But this is a simpler and could be more inaccurate method. So we don't like it, right? We try to avoid it, yeah, or improve it. Yeah. So then, how we can relate this with the past and the fossils. If we only use present animal locations with climate information about the preferences of these animals, we can indeed do like really good models. Yeah. But if we have access to the fossil and past information, what we are actually collecting also is preferences about the climate that these animals were enjoying or tolerating. And in that case, and in some cases, can improve our prediction of distribution or our map of the preferences about the climate that this species is preferring. Yep. Well, now I'm going to leave you with Rod, that is going to talk about the awesome biodiversity here in Australia.
Here we go again. Sorry, guys. Me. Thank you, Juan. Yeah, so I don't know if you guys know, but Australia is the land of lizards. So we have over 1,000 species of reptile species here in the country, but just with lizards, it's over it's over 800 species of lizards. That's just insane. That is a lot. And what that brings us is to have all these different kind of morphology, like body shapes and adaptations to live in different habitats. So we get from limbless lizards, like no limbs. Due to evolution, they lose it. We will talk about it later. But then we have arboreal species, grandulars, these beautiful geckos. Geckos are my passion. And and they all occupy different habitats because they can. And why they can? Because they have different morphology, different attributes, physiological and morphological attributes that let them do that. So if we think about, when I talk about differences, like let's talk about geckos. The next two slides are geckos. So these guys have such a different toe difference, toe difference. And so it depends on where they live, what they need, what they have, and what they keep. So some of them have toes with claws. Some of them have toes with digit pads. That's what we call. And some other differences come in tail, for instance, tail use and shape. So that is Mike's hand with a marble gecko from Hong Kong, I think it was. And so that gecko stores fat in the tail, but gecko mostly stores water in its tail. So there's so many differences even within big taxa groups. And to the point where we just lose toes and legs and everything. So that's how probably you imagine in the top left, is that how probably you imagine a skink, like a slender guy with 4 limbs and 5 toes in each limb. But then we have like skinks that don't even have like the front limbs at all. And then we have skinks and geckos that don't have either. So how does it work? Well, they slide. So it's where they live, what they need, what is going to be useful for them. And then we have all these different, like currently we have all these different amazing species of snakes and frogs that I will show you later. And from here, how you say, back, right, yeah, right bottom. In the right bottom, we have like sea snakes, and then there are some other snakes that are arboreal, or they live in the desert, or venomous, non-venomous. It's just such a huge variety, as well as it happens with frogs. So we have over 280 species of frogs. We even have a burrowing frog here in Australia. So it's just so amazing how diverse they are in their morphology and where they live, what they need. And so, okay, so how all this diversity happened, how we came up with all that different kind of species. And so lucky for us here in Australia, we have this just colossal quaternary museum fossil collections. Like, it's just so big. And this is the first study that is going to utilize these collections in order to match the past, the present, and be probably able to predict in the future what is going to happen with species and the environment. Like that. So just to show you a little bit of the different fossils that we have, because I show you the current species that we have, but look at that, like that tiny top left, that is a tiny jaw of an agamid, a little dragon. So from that tiny kind of stuff to osteoderms of goannas. Osteoderms are the bones underneath the scales, that is the skin of the goanna, the varanus. And in here we have an example of a snake vertebra. So we have everything sitting there for us to go and study it. There. So I will leave you with Beck now. And just to remind you, what are the 3 methodologies we are using in this project is the CT scanning that I would talk later, ancient DNA, and no, I don't leave you, but yeah, I keep talking. Sorry. I think I just talk a lot. So these are the 3, sorry guys, so these are the 3 methodologies we're going to use. And CT scanning, ancient DNA, ecological modeling. And now I will keep talking and tell you about CT scanning. So CT scanning, well, that's me really happy there because that was the last tube that I scanned for a project back in the day with, scanning 1,000 fossils for a very short project. And after that, we developed a technique that we published. I will tell you later. But yeah, so this is the size of that is a gel cup. That is the size of the fossils we deal with from the quaternary collections. So CT scanning, what is CT scanning? Well, CT stands for computed tomography, basically X-rays. And so the beauty of this technique is that it is a non-invasive non-invasive technique. So I can take anything out of the collection, the most precious specimen in the collection, and I can CT scan it and not cause any damage to the sample. And that is really a really important fact when you're when you're doing experiments or applying techniques to specimens in a museum or preserved or specimens in a project, because you want the less damage possible. And so this is a beautiful technique to get to know from the outside to the inside of a specimen. And so basically what we do is I put a whole bunch of fossils, and I will show you later how, but in that falcon tube that is standing there, that on the left, I have a falcon tube full of 90 tiny fossils there. And then an X-ray source hits the tube in 360 degrees. So one of the most important things when you're CT scanning is try to keep the sample stable, because if it is a micro CT scanner, the sample is gonna rotate in order to get all those projections, the X-rays to hit the sample in every single angle. And so it is important to keep them steady, otherwise you get blurry and low quality scans. And that was a challenge, let me tell you. So scanning so many at the same time. So basically, yeah, the sample rotates and the source hits the sample over 360 degrees. And then with a reconstruction algorithm, visualization, 3D visualization software, what we do is reconstruct the sample so I can get a full animal or a full set of fossils, and I can visualize it in 3D to analyze later. So this is basically how that white falcon tube looks as soon as the X-ray hits it. So inside I have all those fossils. This is an example of how do I see it afterwards. Well, these are osteoderms. The other ones are jaws, but you get the idea. So like, this is how I see it afterwards in the 3D software. And this is an example on how I can play around and spend a lot of time making Australian colors for Australian fossils. And this is just beautiful. So we can basically remember what we are trying to do with the CT scanning part of this project is to compare shape and size. And this is a great way to extract those images to analyze it. And so this is a technique I was telling you about before. So on the left, we have the publication we did this was the first publication that appeared like with an innovating technique on how to scan microfossils, a bulk of them at the very first, like at the same time, sorry. And then we are trying, this is a picture on how to like just to scale how small they are. And that is one of my biggest fossils. So to handle them is quite a challenge. But now we are developing a new technique with Queensland Museum to refine our previous technique. So stay tuned because it's going to be really cool how we are doing it at the moment. And so 2 more things. So what can we do with the CT scanning? So I said, I want to compare shape and size. Okay, so size is a metric. It's very straightforward measurement, right? So from the scans, I can measure them, linear measurements, other kind of measurements, that's fine. But how do I compare shape between an animal that lives in, that is a tree climber and a glandular? Because the shape of the body, as I showed you before, it changes. How do I compare shapes? Well, so we grab those 3D images and we put them into programs like MeshLab. I'm not going to go in depth into the methods that we use, but I will tell you that it's called geometric morphometrics. And basically, yeah, we can quantify the difference in shape. And that's how we will be able to compare the fossils that we scan. Oh, yeah. And oh, yeah, that tree was with mine too. In the middle where I have the thorny devil. So another thing that we can do is to 3D print. So afterwards, well, I'm happy to show you, but so this is the scan of, if you see the figure on the left, so that little dragon, I scan it. This is the second picture. The third picture is a smaller version of this, and this is a bigger version of the same. So I can come up with a tiny, tiny specimen, blow it up, 3D print, and have fun. But also, it's a very good resource for our outreach activities like this one, because people can get to understand how versatile CT scanning is. And I can tell you straight away from this that this guy is nocturnal, this guy is diurnal. So this one is most awake at night. This one is active. This active period is during the day. And come and ask me why. And yes, I'm done. So now it's time for Beck to come and remind us what is going on with this project. Thank you.
Okay, hi everyone. So Ro has given us a really detailed look into the amazing CT scanning technology we'll be using in this project. And I've put this slide up again just to remind us about the main questions underpinning this whole project, which are: how has Australian reptile and frog biodiversity changed over time? How is climate involved? And what about the future? So meaning how we might use the information we find to make future predictions involving reptiles, frogs, and climate change. So of the three main techniques we're talking about today, we've already covered the CT scanning now. I'm going to cover the ancient DNA, and then Juan will come up later with the ecological modeling. So over the next few slides, we will talk about ancient DNA. Strap yourselves in. Okay. A lot of people don't realise that we have a really thriving sciences department here at Melbourne Museum. We've got some amazing researchers studying all sorts of things, including fish, mammals, birds, reptiles, marine life, insects, dinosaurs, the list goes on. We even have several custom-built lab spaces in the building, one of which is our molecular lab, where we do all of our genetics research. And that's what you can see in these pictures here. So you can also see that we have a really spectacular view of the Royal Exhibition building in that left image. So it is a really lovely space to work in. Now, most of us probably have a good basic understanding of genetics and what DNA is, even if it's just from high school science or watching CSI Miami, which may or may not be very accurate. At its most basic level, we can explain DNA as being a molecule that's inside the cells of all living beings that provides different codes to instruct the body how to make us look or develop. So, for example, DNA has a specific gene that codes for hair colour and another one that codes for hair type. So for me, I've got brown curly hair. If I shaved my hair off today, it wouldn't grow back as straight and blonde because my DNA has a code that says brown curly hair. So that's what's going to grow back again. All kinds of traits that you can see are coded by our genetics, things like eye colour, skin colour, how tall we are, and even traits that you might not be able to see, things like allergies or medical conditions. So what is ancient DNA? Sounds pretty fancy, but it's actually the same kind of DNA that I've just been talking about. It just comes from a different source. So when we talk about ancient DNA, we're talking about just coming from samples that are really old. So in our project, we're going to be looking at fossils, which can be tens of thousands of years old, the ones that we're going to be looking at. So it's actually pretty cool to know that we actually have the technology to take DNA out of these really old specimens and use the data in our analysis. So it's pretty cool stuff. So what can DNA help us with? Well, DNA asks all sorts of questions about the world around us. We can use it to identify what species we might be looking at, which is important with some of these fossils that we might not have any other way of knowing. It can help answer questions about the evolution of a species, and it might also help in answering questions surrounding conservation, which is really important when we're working with rare and endangered species. So in basic terms, we can answer a lot of these questions by looking at different DNA gene codes for each specimen and then seeing how similar or dissimilar it is to another specimen. So how do we work with ancient DNA? Ancient DNA is actually quite different to work with than DNA that's been taken from a present day specimen. And this is because in most cases, the quality of the old DNA isn't so good. So it makes sense. They're old fossils, they've degraded over time. The quality's just not so good. So over time, scientists have been working on this problem and they've come up with a few solutions. So there's a lot of special methods and techniques that have been employed to be used for ancient DNA work. One of the biggest things is actually having a really special lab environment to do the ancient DNA work in. And the biggest things around this are cleanliness and a lack of contamination. So the last thing that we want is for us to be accidentally amplifying up DNA from ourselves. So a little piece of skin or a bit of hair. That's not what we want with ancient DNA because it's really hard to actually get the DNA out of. So we want to make sure everything's as clean and sterile as possible. So you can see in this picture here that, as we're all pretty well aware of these days, full PPE is pretty much what's needed in a DNA lab and just really strict hygiene, cleanliness techniques. If we come back to our project, so specifically our ancient DNA research on reptiles and frogs, we're going to be using fossils from that really big museum collection that Rose spoke about earlier to extract DNA from a number of different species. We want to use those ancient DNA results to help answer questions about whether reptile and frog biodiversity has changed over time and space. And from this, we'll be able to look at where different species fit into the evolutionary tree. We may also be able to add this genetic data to our larger data set and explore links between things like species diversity and perhaps even climate change. So let's take a step away from the genetics now and look at the very starting point of this project. For us, this has been in the form of building a database about what we currently know about the reptile and frog fossils in Australia. There's quite a lot of published papers out there, but if you think about the large amount of fossils, some of those pictures that Rose showed is not even the starting point of what we already have in the collections. There's actually very little research that's been done. So that's why we're actually really excited by this project, because there's so much scope for us to actually fill in so many gaps in the knowledge. We're using this database to help keep track of where the fossils are as well. So it's going to be a really good resource, not just for us to use to be able to locate different fossils, but also for other researchers in the future. So we are hoping to publish this database, and then it's going to be accessible for everyone to use if they want to access the fossils for other projects into the future. And now I'm going to pass on to Juan, who's going to talk about ecological modeling.
Well, so far we have explained a bit of 3D fossil scanning, ancient DNA, and all of that with the objective to identify fossil specimens to the species levels. So now it's time for modeling, but not the modeling that we can see in Milan in Italy, but statistical and mathematical modeling, right? So to do that, we not only need like fossil specimens, but we need also current species. And in this case, we're going to be gathering our record from two sources. One from Atlas of Living Australia, what is like online biodiversity database that is public and everybody can access, and encourage you to do it if you haven't done it before. You have a lot of biodiversity information about Australian species in it, and you can collaborate as well with them. And the other source is through field work. And in this case, but I think I went too far. In this case, we will be doing like field work in poorly sampled areas around the east coast of Australia, specifically in 3 caves and in their surroundings. So one of these cave areas is in far north Queensland. You can see here is an isolated area with big escarpment, difficult to access. Therefore, there's a lot of information that is hidden there that we are looking forward to gathering. And definitely it's going to be an adventure. Then how are we going to cut those species? From trapping to photography. Here I can show you some photos about the methods that we are using. Here on the left, you can see me setting up a fence that will be used to redirect the reptiles that hit the fence toward the buckets, and then we can collect them from the bucket. Or my colleague on the top right setting up funnel traps that is like a tube with a small hole inside, and the animals can come in, but they cannot come out. And then we're going to process them. When we process the records, we can use those records from current species and climatic information to fit our statistical models. And then these statistical models, along with past, present, and future climate models, ultimately can give us or render like spatial predictions about the distribution, past, present, and future. But what if we include the fossil from the species that we have found before? So when we do this, we can add more records to the cluster of record that we had before, and we can improve our models, and therefore we can improve our spatial predictions as well. Another type of modeling that we will do is a mathematical modeling using current and fossil records. And the objective of this is will be to understand how the community has changed from past, present, and future in these 5 cave sites. So let's imagine that in one side we have this assortment of species, and we look back at one of the cycles from the ice age that we were talking before, cold and dry, for example, and desertic area. In this sample, we're going to have one type of assortment of species, let's say these 5 species, and in a different climate cycle, we will have a different assortment of species. Some species can remain, some species can vary. But at the end of the day, basically, we're giving information for conservation managers about how the community has changed over time. And we can use that also to predict how the community will be in the future for predicting try to reduce the impact of climate change on that community specifically. Yes, this is the end of the techniques explanation. But Beck is going to summarize everything up for you right now. Yep.
Little bit of back and forth tonight. Okay, so I'm going to wrap up the talk by once again just reminding everyone of what we're ultimately aiming to do with this project. We're really interested in finding out as much as we can about how reptile and frog biodiversity in Australia has changed over time, how climate change might be involved, and whether this information can be used to help make predictions into the future. This project is the first of its kind to be using some very innovative technologies like CT scanning and combining a really wide range of scientific fields, such as paleontology, macro-ecology, tomography, which is a CT scanning, taxonomy, and genetics. And once again, we really want to shine a light on how climate may have impacted reptiles and frogs in the past, in the hope that this information will help guide us into the future with issues surrounding climate change. This project has so much significance to the scientific and wider community. It will be the first study of its kind to really explore Australian reptile and frog biodiversity patterns over both time and space. We'll be highlighting the many ways that museum collections can be used now and into the future. We'll be making use of new and emerging technologies, which will hopefully help other researchers interested in using these techniques in their own studies. Any discoveries we make for endangered or rare species can hopefully be used by decision makers to help in conservation management strategies. And this study will provide some really exciting research opportunities for students to be involved in, like one. We'd like to thank you all for coming to listening to this talk, both of you here in person and those that are listening online. I know we have people streaming from all over the world, so we really appreciate you coming to hear about this exciting research we're undertaking here at Museums Victoria. If you have any questions or queries that we don't get to answer tonight, please get in touch with us. We have some contact details up on the screen, and you can always just get in touch with Melbourne Museum through the generic contacts on the website, and they can direct the questions to us specifically. Thank you all again.
Thank you. Look, some fantastic science there and some amazing images. So please give the speakers a big congratulations just on the presentations alone. As I said, we're going to take some questions now, and Kate Phillips is also taking some online as well. Kate?
Yes, I've got a question from our online audience, if I can jump in first. We have someone who's asking whether your work uses information from the national collections that are held by CSIRO.
Say that again. Yeah.
So the question was, will your work use data from the national collections that are held by CSIRO? I imagine that those are all fed into the Atlas of Living Australia, that information, but would you like to add to that or comment on that?
Yeah. I can answer from the CT scanning part of side. Then you can just probably explain. But yeah, the collections we are going to work with are basically Queensland Museum collections, and then South Australia collections as well. But yes, it's basically the quaternary collections that we have in Queensland Museum for the scanning of the fossils.
Yeah. In terms of the species distribution model, as we saw before, is like basically Atlas of Living of Australia and our own work. But as long as those collections held by CSIRO are like uploaded in Atlas of Living Australia, definitely they will be there, as long also as they have enough accuracy in the records. Yeah.
And I don't know if this one's on, but just to add to that as well, we will be, as well as the Queensland Museum fossil collections, we'll be using collections from Melbourne Museum here, as well as some of the other museums around the country, including South Australian Museum. But we're starting with the Queensland Museum collections and the Melbourne Museum collections.
Great, thank you. And I do have one more quick one, which is that if people are, would like to get involved in looking for fossils themselves, is there any way that they can be involved in this sort of work?
Oh my God. I mean, thank you. So, because I wanted to put these in my talk, but I didn't have the time to do it. So I went like, oh, maybe somebody wants to help us. And so that is going to happen. Yes, next Saturday, actually. But usually we have like what is called a fossil open day up in Queensland in the Capricorn Caves that I showed you before. And that's where we collect all the fossils from in the Capricorn Caves area. So from the northern caves, that's what it usually happens every year. Because of COVID, it didn't. So now it's part of a big event called Capricorn Caves, Big Science Adventure. And so people can go there. As you can see, we have whole families like searching for fossils, and that guy there found that little one found like a very important fossil, an amphibian fossil for us. So we are using it in our studies. So yeah, this is great. So jump in the Facebook announcement from Capricorn Caves if you have the chance to go there this weekend. That is even amazing. Otherwise, yeah, you know that is there for next year. Oh my God, so cool. It is really fun for families.
Thanks, Rosie. Other questions out there? Anyone in the audience? Yes, Alice, stand in the front here.
Thank you. I wondered if you could just tell us a bit more about the timescale of the project. Like, how far through are you? How long is it going to take? That sort of thing.
Yeah, that's this one. Yeah. So it is an ARC discovery project, so it's funded for 3 years. We have had some delays because of the obvious COVID. Let's not say the C word, but unfortunately we do. So we're about a year in, but we're a little bit behind because we've been restricted with accessing laboratories. So we're starting to now amp things up, and we're just about to start getting into the CT scanning side of things, which is the first step. So what we have been able to do is work on, I guess, the database side of things and get that that information about what's already out there under our belts, and now we can really hit the ground running with all of the actual practical work. So the CT scanning is going to be starting soon, and we can also start the genetic work quite soon. Juan is already got a full calendar for his PhD with field trips and all sorts of things. So in terms of timescale, technically about a year in, but we're not quite. We've had to get a bit of an extension. So it is hopefully going to be run over about 3 years, a little bit longer.
Any other questions out there?
More from now.
Yep. Okay. Great.
Online audience. One of them is how can students get involved in this sort of work?
Well,
Yeah, the steer on that.
yeah, well, some students could get involved in this work, doing it as I did it as well, like applying for a PhD. There are actually one open for application. So if they want to talk with us like later on, yes, that could be okay. Then in the field work, we always get like people collaborating. And even when still don't know how many people can collaborate, and then some of the field work you saw is in Chilago in far north Queensland. So if someone is around there and is interested in collaborating, they can let me know. And we can consider it, because definitely we had to be like few researchers with training, but also people that can help with other things is always helpful, definitely.
And just to add to that, as Juan mentioned, we do have a PhD position open, which is exciting. We also have some honours positions. So if there are people who are in that stage of their science careers, they're just going through undergraduate, they're looking for honours projects, please get in touch. We'd love to hear from you. Feel free to just contact the email address that we had up, get in touch with the museum, get in touch with Jane Melville because she'll be supervising you, and we can chat.
Yeah.
Would love to hear from potential students.
Fantastic. That's great news. And another question, someone was really obviously their curiosity was piqued by your diurnal versus nocturnal. How can you tell whether they're active in the day or the night just by looking at their skulls?
basic physiology and morphology together. So if you, how can I show you this now? So if you see here, there's a hollow there in the middle of the skull. I don't know if you can see it or not. I can show you later, but
that one right in the centre, is it? That are you talking about the in the centre of the skull?
Yeah. Yep. So that one there, that is called pineal foramen. So is where the pineal eye sits underneath that, and that's what it tells the animal if it is night or day, and who will need it. Who needs to know if it is night or day most that these guys that are diurnal and they need to get active as soon as the sun comes out and the temperature is accurate for them to do it. But this guy is a nocturnal gecko. So it's not an Australian species, sorry. It's an Asian one, but that's okay. So this guy doesn't have that because he doesn't care. He comes out during the night, and during the day he lives in crevices, Cirtodactylus pulchellus, and this is Rankinia yemensis. So Australian, Asian, but that is the main thing, that as soon as you see this, you go like, yeah, diurnal, he needs to know when it's the day, nocturnal, he doesn't care.
And also, I presume, reflecting of living in a different climate, what may be a warmer climate where you can be active in the nighttime.
Yeah, that's correct. There's still like, I've done my bachelor's thesis with a southernmost gecko in the world. So, and he's nocturnal, but and he lives in Patagonia, Argentina. So it's just a little bit of, there are many rules and a lot of exceptions to the rules, but this is an Australian gecko, Nephrurus levis. So he's nocturnal. So as you can see, he doesn't need that either. So it's pretty.
So if you know what to look for, you can see these wonderful patterns.
Yeah.
I think we should be selling these in the museum shop. Just make them slightly bigger and you can have a Halloween mask or something there. So, yeah. Any other questions there, either online or Alice? Yeah.
Sorry, I had another one. Could you tell us a little bit more about the when you're doing the DNA amplification from the fossils? Does it matter like which bit of the fossil you use? Are you looking for teeth over bones or?
Yeah, so what we're going to be focusing on is the vertebrae, so the backbones, because apparently they're the easiest ones that we can actually get DNA out of, and the jawbones as well. So that's going to be our focus. We haven't started yet, but that's what our plan is. And the fact that we just have so many of those types of bones in the collection. So we've got lots to work with, and they are very good at getting the DNA.
Yeah, those are the parts of the body that get preserved better because they are denser. Yeah, they are more dense, and also kind of like pieces of leg, pelvis, vertebra, and jaws. Yeah.
So when it's amplified, does that mean you're better able to compare it to DNA from a current animal? Is that why you amplify it?
Yeah. So we'll be able to amplify the sequences. So look at a specific region of a gene and then compare it to things that are living today, so current species, and see whether it's the same species or a similar species or something completely different that maybe we haven't seen before.
Cool.
It is cool.
I could keep asking you questions about that, but I'll stop.
Any more questions there? Yeah, one up the back.
I just had a question on, you were saying with the ancient DNA, in the current collections of like fossils that were collected 100 years ago or 200 years ago, is there a problem with using or getting DNA out of those earlier things because of the way they were stored or collected or preserved over the 200 years?
Yes. Yeah, right? So that is a potential problem. that we have. Luckily, we have a lot to work with. So we have that flexibility of if one specimen doesn't work, we can maybe choose another. We also have an option of looking at something called proteomics, which is looking at, I always stumble over that terminology, which is actually looking at proteins instead of the actual DNA. So we kind of have that up our sleeve a little bit because we know that we have that problem with a lot of the Queensland specimens. They've done a few sample runs with the ancient DNA and they know there are some problems with bones that have been degraded over time, and it's just really hard to get the DNA out of them. So that's a part of the project that we didn't talk about today that we've kind of got up our sleeves if we need to go there, but we're hoping that we've got enough samples because we just have so many fossils that we'll have enough to work with the DNA. But it's definitely a potential problem.
Yeah, and another problem that can happen with all collections is like many times they don't know from the layer that they were collected. And the temporal margin is so big that probably we cannot use it, even if we know which species it is. That's gonna limit the use of it. Yeah.
Thank you.
Any further questions? Any more online there, Kate? Yep, here's one.
Hi, yeah, I was just wondering, like, because it sounds like such a crazy undertaking, right? But are you guys focusing more on like specific species or like what's your process as to deciding how you guys are gonna?
For the DNA work, yes, we're not going to be doing every species. We're going to select certain species. So we'll sort of have, a couple of lizards, a couple of frogs, a couple of snakes.
Yeah, for the CT scan part, it's kind of, we do have a lot of samples of skinks, dragons, geckos. Geckos not that much, but skinks, dragons, snakes. That is just a big broad from what is preserved. So yeah, we will tackle that and as many species as we can work with, but in those main big groups. Yeah.
So it's kind of like working with what you guys already have and then just finding out whatever you guys can find out.
What we already have in terms of the collection, like the fossils. Yeah. So we are not gonna go and extract more fossils unless we really have to, but we don't, because it's kind of we have hundreds of trays, as I showed you before, sitting in racks at the museums. So we have so many to work with, but the full preserved variety goes around those groups. Yeah.
And when Ro talks about those trays, most of them haven't been sorted. So they're literally trays full of fossils mixed with dirt, mixed with everything. So there's a lot.
Okay. Most assemblages in the caves tend to be skins, just basically because they are more abundant as well. So, and also a snake vertebra.
Yeah.
And for a species distribution model, we will be doing like as many reptiles and frogs as we could, but we had to select out of the quality of the data that we get. For community assemblage, we will be focusing on skins, basically because of that, because in the case, most of the species that we can find are skins.
Okay, that's interesting. Thanks.
Great job with your talks. So with fossil records, you've always got to worry about bias. So sort of my question relates to what you were just talking about. So what sorts of biases do you get in these reptile fossil assemblages, and how are you going to deal with that when you're trying to compare them to communities that of the present day or the future?
Yeah. So, well, in terms of species distribution model, we are adding the fossil record to the current records. So it's just like adding a bit of more information. So we're not gonna end up with the full bias of like not having records from the same time, right? For predicting the distribution, right? Because we can find this type of bias. Basically, it's like temporal bias. The other is a spatial bias because you don't know if the fossil has been, well, you had to know about the taxonomy or how the fossil has fossilized to understand where the fossil or the animal is coming from. Yeah. And in terms of the species distribution model, we are kind of like removing a bit of the bias, or most of the bias, doing it like that, just incorporating fossils from those specific time and the climate information from that specific time. So we will get climate models for that specific time again. Yeah, and then we will get the bioclimatic variables exactly. So in that case, what we have is like a species that was proved that is the same species not long time ago, with no much modifications or adaptations with information about the climate that it was liking. Yeah, I think that is the mainly idea for the community assemblage model. Is still to be discussed. Yeah, it's going to be the last part of my PhD. Yeah, we got definitely we thought that about that.
You're going to do a lot of field work and catch some lizards, right? So can you compare the assemblages of what you find today, or in the present day, with the recent fossil record in those caves and get an assessment of bias that way?
Pardon, can you repeat it? Yep. No,
can you use the data you'll get from your fieldwork and compare that to the recent fossils in your caves to get an idea about what bias there is of what's falling in the caves compared to what's on your site? So
that's a good idea.
It's a great idea, Mark.
Yeah.
Okay. Any further questions? Anything online, Kate? Okay. Thank you very much. It was a wonderful talk. It's Science Week, and I think this talk and this project actually highlights that science is a creative endeavour. Rebecca put up a slide earlier which outlined the various components of this project, and even imagining this project is a very creative process, but it involves paleontologists, zoologists, ecologists, and then it brings in all this wonderful technology and techniques, genomics and the CT scanning. These things weren't applied to museum specimens 30, 40 years ago. And I always think that when some of these specimens were collected, and I think Rocio, you mentioned being in the fossils that have been in the collections over 100 years and so forth. So the first curator of zoology was appointed about 160 odd years ago, and he collected many things, but he couldn't have imagined how those specimens would become important later in time. He knew nothing of DNA or genomics or any of those things, and now we're extracting DNA from specimens that were collected 150 years ago that are helping to inform conservation strategies for vulnerable species. So, as I said, it was a wonderful talk. It brought together many different disciplines within science. But imagine, when in 100 years' time, if you were sitting in this audience, who knows whether technology is going to take the science, but also that the museum specimens and collections will still be that important research infrastructure. And that's one of the great purposes of museum collections. So please, once again, thank our three wonderful speakers. I'd also like to thank our technicians up the back, making it all work for us. Visitor experience officers who helped get people into the building and into the lecture tonight, and all those behind the scenes. All those behind the scenes who help host this event. And especially including the MV Lectures team, Kaz McClellan here, Moya McFadgen, I did see Moya before, and also Kate Phillips over the back here. So thank you very much for your help.